As we all know that across the world population, all are being affected by a very small virus that's not seen through our eyes, and it is known as COVID-19, as the name has been suggested because it was found in November 2019, and it is also known as a class of coronavirus. At this moment, I have already made an attempt to gather some information regarding the virus, how the virus is, and how common or general definition of the virus and the cause of it. Let's begin with the slide. So COVID-19 basic description. So let me introduce you to the viruses. That is, coronavirus are large family of viruses. They cause illness. That is, we know that we get sometimes common cold, and also from common cold to more severe diseases such as Middle East respiratory syndrome and severe acute respiratory syndrome. So in the short form, it is known as MERS, MERS and SARS. So 2019 nCoV, N is the new COVID strain. So it is a new strain that has not been previously identified in humans, and we have not ever faced such a big or difficult times in the recent years. And it is known as COVID-19 or coronavirus disease. So before going to the coronavirus, we need to know what is a virus. So let's understand the viral definition. So a virus is an infectious agent. It's a small in size and it cannot be looked into by our naked eyes. And it has a very simple composition and it can multiply. The major cause of multiplication is it requires a host, that is a living cell of animals, plants or bacteria. If it doesn't have a living environment nearby, it will not survive. So it will stay without any multiplication or any process of the life cycle. So you can see that virus is a very small, minute organism. And there are different classes of viruses. It's not only corona, there are a lot of viruses like flu, common cold, HIV, influenza, etc. So let's understand where do these viruses come from. So each virus has a unique origin, that is, the virus might have been linked to the previous viral link or it will be different from the original species. So a species of virus can be different from the other one. So it is a rare case that you can see that the virus jumps between the species. If a virus successfully wants to jump from one species to the new species of its host, then it needs to do a lot of things. So that is, it must establish an infection in the host site. Host means it might be a human being or an animal. So for example, like a virus has been attacked in an animal. So the virus keeps on attacking the same species of the animal until and unless it finds a new host or new species. But it is a very difficult task for one or virus to establish a new infection in a new host. So what are the steps that the virus should take itself before it establishes an infection to a new host? So it must be able to find a new host. It should attach itself to the host's surface membrane, and you can see that it should be able to multiply there. The virus may also be able to replicate itself, so it should be able to replicate itself to infect others and transmit itself to them. That is, it should have the ability to transmit its proteins to the host membrane. For example, influenza virus, H5N1 or bird flu can infect humans from the birds. So let's understand now, how do these viruses get their names? So viruses are named based on their genetic structure to help to facilitate, that is, it helps to facilitate the scientists or other researchers to help them to name the virus and also the cause of the virus. Once the regular disease mechanism of a virus is known, it can be classified under a list of viruses that is already existing. And if the existing strain of virus is already known to the scientists or researchers, they will be able to develop a lot of the similar diagnostic kits or vaccines or medicines for the human purpose. So who are those who name the viruses? They are the virologists. They study about the virus description, diseases, and how fast they are going to replicate. And wider scientific community scientists also do the naming ceremony of the viral species. So viruses are named by ICTV, that is International Committee of Taxonomy of Viruses. So virus can also be classified under taxonomical classification, that is under realm. So you might have seen that taxonomical classification has genus, species, etc. But it is a higher end classification of microorganisms or organisms. It is higher than the kingdom. So you can see that coronavirus, that is, I'm speaking about coronavirus, it is still unranked virus. So what is the realm? Realm is Ribovira, kingdom is Orthonavirae, phylum is Pesoviricota, class Pesoniviridae, order Nidovirales, family Coronaviridae, subfamily is Orthocoronaviridae. Classification of viruses, as you can see in the picture, that some of the viruses are enveloped and some are single-stranded RNA virus and some of them are DNA viruses. So DNA viruses are easier to be combated with certain medicines or vaccines, but single-stranded, some of RNA viruses are more virulent than their counterparts. So you can see here that coronaviridae structure is given. Then you can see a lot of different viruses. So retroviridae, orthomaxiviridae, flaviviridae, etc. So next we will see, so to understand a virus, we already have learned about the viral definition. So next we will understand the structure of RNA virus. So coronavirus has already been told by or concluded by the scientists that coronavirus is RNA viral species and it is also a single-stranded RNA virus. So a RNA virus is a virus that has ribonucleic acid, that is RNA describes for ribonucleic acid as its genetic material. So what does this nucleic acid do? It is usually a single-stranded RNA. So S is RNA, represents single-stranded RNA. So RNA viruses can also be double-stranded RNA. But you can see that coronavirus is an RNA virus which is single-stranded. So what is RNA composed of? So RNA is composed of mostly sugars, that is ribose sugar, phosphate residue and a purine or a pyrimidine. So purine and pyrimidine are the base pairs. So you can see in the structure behind that adenine, uracil, guanine and cytosine. So let's understand how do RNA work. So RNA carries information of amino acids sequence. So amino acids are present in the proteins. So you can see that RNA carries information of amino acid sequence from the genes. Genes are hereditary materials. So from the genes they carry to where proteins are assembled in the ribosomal site in the cytoplasm. So which type of RNA does this process? It is messenger RNA. So you can see the structure. It is a general structure of a bacteriophage. Bacteriophage is a virus that infects the host. So you can see that it has a head-like structure. It's protein coated, and inside the head-like structure there is a nucleic acid and also the presence of DNA. So in the influenza virus, you can see that there is a RNA instead of the DNA. That is the difference here, and membrane envelope. So now a question might arise that do humans have RNA? Yes, of course humans have RNA, but the DNA will help the RNA to synthesize the further protein molecules. So you can see that in humans we have 4 kinds of RNAs, that is ribosomal RNAs. We make RNA copies of our own DNA genes. So in the structure that is in the figure, you can see that in the left hand side you can see the deoxyribonucleic acid, that is DNA. It is double stranded and it's also known as double helix. So you can see 5 prime is at the top, 5 prime to 3 prime end, and 3 prime to 5 prime end is wound DNA structure. So it also has base pairs AUGC, ATGC, that is, you can see that it also consists of sugar phosphates. So the only difference, you can see that here double structure, and it is a single stranded structure in RNA. So you can see in structure of DNA the base pairs are ATGC, for RNA it is AUGC. So where is RNA found? RNA is mainly found in the cytoplasm of the cell, though it is synthesized in the nucleus. So usually RNA is being synthesized in the nucleus, but it is found in the cytoplasm. So what are the roles played by RNA? So we have known that RNA are of 3 or major kinds, that is messenger RNA can be shortly called as mRNA, transfer RNA shortly called as tRNA, ribosomal RNA, that is rRNA. So let's understand one by one. That is messenger RNA. What does it do? It carries genetic information copied from DNA. From the DNA, a messenger RNA copies the genetic information in the form of 3 base code words, that is, I have already spoken about the base pairs, that is ATGC and AUGC. So in the 3 base codes, that is ATT or AUG, etc., each of which specifies, that is 3 base pair codes specifies a particular amino acid. So transfer RNA. So transfer RNA, what does the transfer means? It's helping to move the molecules from one place to another. So what does this tRNA do? It brings amino acids to ribosomes during translation. So what is ribosomal RNA? It is actually a form of structural components of protein making structures known as ribosomes. So ribosomes are the site of protein synthesis. So you can see the picture here of messenger RNA, ribosomal RNA. So you can see transferred RNA. So you might be thinking now, is RNA dangerous? So if RNA is dangerous, then how would be DNA synthesized RNA in humans be? So usually it's not dangerous, but mutant ribonucleic acid molecules can be toxic to the cell. What is mutant or mutation? It is a change that occurs in DNA or RNA. So usually you can see that base pair structures are AUGC and ATGC. But what happens here is that there is some damage that might happen to these base pairs, and it might lose one of the base pairs, or it gets replaced with the other base pair, and there is a mismatch of the particular mechanism, coding mechanism or synthesis of protein itself. So you can see that when the factor is copied as a result of environmental factors, mutation or a change or damage takes place with respect to DNA or RNA, can be by the exposure to UV light, or it can be a cause of pollution, or it can be due to cigarette smoke, etc. So RNA can be dangerous when there is a mutation or change in the particular sequence. Sometimes viruses such as those cause flu, HIV, human immunovirus have RNA-based genome. So viral RNA genomes are much more. See, viral RNA genomes are more prone to mutate than those of the based with DNA. So you might be thinking now that why do viruses spread faster than anything else? So viruses undergo evolution or a change faster because of the natural selection or the habitat it is within. So when two viruses infect a cell, so we'll take an example of two different viruses, and it infects a particular same cell in an organism. What they do is they exchange their materials with each other and they togetherly mix to form a new form of virus with a different property that would involve the property of the first virus and the second virus. So mutation in humans and viruses are two different studies wherein so there is an ancient example which says originally all human beings, initially we all had brown eyes. So then about 8,000 years ago a mutation occurred. I told as before that there is a mutation or a change in the gene that occurred because there is some change in the amino acid 3-word code, that is ATT or TAT or AUG or etc. So the code of tyrosine is TAT. So that got changed to aspergin. So tyrosine base pair was changed to aspergin, that is GAT. So less tyrosine, which turns brown, that is led to blue colored eyes. So there is more tyrosine, there was a chance that the brown colored eyes remains as it is, but it due to the less amount of tyrosine present and more amount of aspergin present, so it turned that in humans there was an evolution of blue-eyed people which see slightly better at night but slightly worse during the day. What is the major drawback for blue eyes are that they can see slightly better at night, but in the daylight it's quite difficult for those people. So amazingly this mutation occurred in only one single human, and all blue-eyed people are related to that particular human being itself. But here in the virus the mutation or change is very faster. So let's take a viral mutation example that was a lab study previously. So you can see here a human flu virus that is infecting the humans, and there is an avian flu virus that affects the birds. So what has been done is they have taken a mixing vessel, pig mixing vessel and human mixing vessel and human flu virus. So you can see in the mixing vessel they have mixed avian flu virus and human flu virus that has been infected to the pig or injected to the pig, and human mixing vessel and human viral flu vessel that was infected or mixed together, so that would cause a major chain in human disease of viruses. So this caused a new viral strain in both pig and the humans. So next what happened was a new strain of virus evolved that would even cause disease to both the pigs and the humans, and it got mutated, and the human population would be devastatedly affected by this process of new virus. So you can see that both the animal and the human will be affected due to the new strain of virus. So why do RNA viruses evolve faster? So what does happen here that RNA viruses are more faster to affect other living beings is that transfer of genetic ribosomes. So ribosomes, where it is used to instructions to build protein. Ribosome is the site of protein synthesis, where usually it happens that DNA passes information to mRNA, that is messenger RNA. From there it moves to the ribosome site for protein synthesis. But it doesn't happen the same in the RNA viruses. So let's understand new coronavirus, which is a positive single strand. So this virus is different from other viruses. How? So it doesn't use DNA, as we had told that DNA sends the signals to the RNA. From there it sends to the ribosomal site, and it's the site of protein synthesis. So you can see that protein positive sense RNA is the same as messenger RNA. So there is a major thing that we should see that in new coronavirus there is a positive sense RNA that will act the same as messenger RNA in the human beings. So once it enters the human system, it immediately acts as a messenger RNA which carries the information of its own viral particles, and it will start replicating in the host cell. So it will translate into protein within the host cell. So that is the major cause that we are suffering today. It will in turn act as host cell protein. So instead of the human protein synthesis, host cell protein starts synthesizing in the human body, and thus it starts replicating in the human beings. So let's understand what is transcription. As I had already told that it is the process by which DNA is copied to mRNA. So DNA is being copied to messenger RNA. It occurs in the nucleus of the cell. Where does this happen? It occurs in the nucleus of the cell. So DNA is copied to mRNA. So you can see in the figure here that DNA is taken off or copied to RNA, and RNA helps in the synthesis of protein. So DNA nucleic acid language to RNA nucleic acid language. So next we'll understand what is translation. So we have already told that RNA copies the DNA. So in this process it's the continuation wherein the information encoded that mRNA has received from the DNA, it is used to, this information will be used to assemble a protein at the ribosomal site. So mRNA is converted to protein. So in the figure again you can see that DNA is converted to RNA, RNA to protein. So major thing is that the information that it received from the DNA, mRNA will use those information to assemble a protein at the ribosome. But it doesn't happen in the coronavirus because it doesn't have a DNA, it's an RNA. So messenger RNA has a single sense positive strand which itself acts as messenger RNA. Once it enters into the human system, it itself acts as the host cell protein. So when it acts as host cell protein, it will get all the nourishment from the human body cell, and it starts replicating itself. So let's see the structure of SARS-CoV-2 virus as per the scientific studies. So you can see the structure here that it has a lot of spikes, a spike where it helps to attach to the receptor host cell. There is a spike protein, yes, and there is an envelope protein. You can see inside there is a circle, and nucleocapsid protein which is present inside the cell, that is in a twisted form. So there is also a membrane glycoprotein. So you can see that it is a virus which consists of a lot of proteins in it, and this protein itself will go and bind to the host receptor. So it is determined by scientists that this virus is made up of spherical particles that have proteins called spikes on them. You can see the extensions here in the yellow color. So these are the spikes on the proteins, or the proteins that will make the virus to attach and bind itself to the human cells. So once it attaches, it will undergo change in its structure. So change in its structure to fuse inside the membrane of the host cell. So then the genes of virus enter into the host cell. So once it establishes an attachment to the membrane of the host cell, it releases its RNA, and viral RNA can only translate and not transcribe. We have already seen the difference of translation and transcription. So you can see that it acts as an mRNA molecule, and it will go to the host ribosomal site, and it will start synthesizing its own proteins for its replication. So let's see COVID-19 life cycle as per scientific studies. So you can see that there is a structure, that is the host cell. It is given in the figure itself, and we have the host cell, that is human beings. There is no other point that this virus can enter than ACE2 site. So angiotensin enzyme site. These enzymes are present at the site of lungs, heart, intestines. So all these you can now understand that why it is infecting directly the throat or the respiratory system and causes the pneumonia, because this virus first attacks the ACE2 enzyme that is present at the lining of the lungs, intestines and heart. So what it establishes a connection to the host cell. First, the virus comes in contact with the host cell's ACE2 entry receptor as given in the diagram. Then its second step is to penetrate. The virus enters the cell body through the ACE2 entry receptor, and cell engulfs the virus. So third is that uncoating. The virus now needs to uncoat itself, that releases its own RNA into the host cell. So ribonucleic acid of the virus is going to get released into the host cell. So what is released now? So the new viral particles are made and released. So if not killed in the process, and if while the release is going on, if it is not killed by itself, then it continues to make new virus by the process of replication. Next is assembly. The new viral RNA that has been produced, these particles are assembled and packaged again. They are ready for going out of the host cell next. So next is biosynthesis, that is RNA replication. The viral RNA enters the nucleus. Nucleus is a specific site where it uses the host cell to create new virus RNA. So once it creates a new viral RNA, it bursts open the cell, and a lot of new strains of viruses that has been replicated will come out, and this will be easier for infecting a large population. So let's understand, we have already seen that the coronavirus or the COVID-19 is going to infect the ACE2 enzyme. So let's understand more about ACE2 enzyme as per the scientists. So ACE2 is known as angiotensin converting enzyme. So ACE2 is an enzyme attached to the human cell membrane as I had told before, of the cells in lungs, arteries, heart, kidney and intestines. So these are actually the enzymes that is already present in our body, which protects our lungs, arteries, heart, kidney and intestines. ACE2 is a central component of renin-angiotensin system. So it's known as RAS system. What does this system do? It controls the blood pressure and regulates the volume fluids in the body. So now you can know that how much pathetic it is to be infected by a COVID-19 virus, as we do not have still a proper vaccine for it, and it is a new strain. So this new strain can enter only through this ACE2 enzyme. There is no other entry point that it can enter into the human system. So let's see, ACE2 is the entry point of COVID-19 virus. So ACE2 is angiotensin converting enzyme 2, which allows the entry of SARS-CoV-2 in the respiratory system, lungs, intestine and kidney blood vessels. What happens when SARS-CoV-2 virus bind to ACE2? ACE2 is the enzyme. So when SARS, that is COVID-19 virus, it binds to the ACE2, so it almost blocks the normal functioning of ACE2 of the host cell. So whatever the function that ACE2 was doing with our human body, the human body functioning itself is affected when this virus blocks the ACE2 normal functioning of the body. It cuts the signaling between ACE2 and ANG2. So what happens here is, once the signals are being cut across the body, it injures the tissues, especially the lungs and the heart. So as per the studies by the scientists as involved, that there are 3 different strains of coronavirus. So type A, actually we have come to know that it was a total different species to species mutation that has happened in this COVID-19 case, wherein it was initially found in the bats and the pangolins. So you can see that type A virus is closest to bats and pangolins, and as I have told previously that this RNA virus cannot jump from species to species very easily. It should make an establishment by itself to the host cell, then only it can jump from one species to another. So let's understand type A virus. Closest to coronavirus found in bats and pangolins, it is the root of the outbreak that is linked to Wuhan, China, and one is almost common in America and Australia. So now let's understand from type A to type B. So variation of coronavirus most common in Wuhan. So there was a total change in the viral description that was infected in Wuhan with respect to the pangolins and the bats. So actually this mutation has occurred from type A virus, and this type B virus was the virus that was infected or affecting the people in Wuhan. So it mutates slowly in China. Actually the virus strain started mutation and it started mutating slowly in China, but it rapidly spread outside China. So we will see now type C virus. Type C is the daughter of type B. So we have already seen that type B virus was showing some of the other symptoms in the China. When it came to different countries, it started showing different symptoms. So type C is a daughter of type B. So one mutation different to parent species or variations that got spread, that is from Wuhan to different countries, it showed its own mutations and different infections, and the type of infections was also different, though the virus was the same. So you can see one mutation different to parent variation. It spread to Europe and Singapore. So till now we have understood that so it was a small attempt made by me to make you all know what is coronavirus, and before that we would try to understand what is a virus and what is a DNA, what is an RNA, and how this virus is making a mess out of the human population, and why the scientists are getting it quite difficult to understand the situation and make the vaccine as soon as possible. It's quite a difficult task because once the virus has got mixed up and it is a new strain and they do not have already the taxonomical nomenclature which was already described in the coronavirus species, so it's quite difficult to understand what vaccine would go well with this virus. So now we have understood that this virus gets mutated from animal to human being, but it is still a big question in the scientist team and researchers team, because as I told before that these viruses take a lot of time to mutate from one species to another, and this is still a mystery. So researchers and scientists are doing their best to get their solution for current situation to make it better. We wish to get a preventive vaccine and completely curable medicine for the whole human race to fight against the deadly infections soon. So I had used some of the terminologies. Let's understand that. Just a recap. So MERS, SARS, that's SARS, ICTV. So virology, taxonomy, evolution, natural selection, RNA, DNA, ACE2 and RAS system. So I wish to tell you about some of the important points or important ways that we need to follow or protect ourselves from this coronavirus disease that has been instructed by World Health Organization. So wash your hands. So why do we wash our hands frequently? So we need not get ill. So it's not only for the virus or any other diseases we need to wash our hands. So after coughing or sneezing, when caring for a sick, before, during and after your preparation of the food, before eating, after toilet use, when hands are dirty, and after handling animals and animal waste. So I thank you all for sparing this time with me to know about viral definition, descriptions and coronavirus and the state or the phase we are going through. Hope we will get over with this tough phase soon, and hope you all will stay well and stay healthy by washing your hands, keeping well hygiene maintained and maintaining social distancing. Thank you all for listening.